So with this video, we're going to go over the slides for chapter 6, and hopefully you're following along with my notes and working on your study guide and stuff while we go through this. This is meant to take 2 lectures if we were meeting in person. So let's get this going here. So the chapter 6 is your first organ system. So we're looking at the integument or the integumentary system. So you've got a set of learning outcomes here as usual. So first, just going over what are the functions of the integumentary system. I think some of them are pretty obvious, but there's some other ones there that you might not have thought about before. Okay, so let's go over this. So first of all, hopefully this makes sense that it's protecting you from dehydration. So it's basically separating you from the outside, even though you're interacting with the outside. It is protecting you, and I think protection is probably one of the first things that you think of. And we're going to talk a little bit about how that works. So it's protecting you from injury, right? And obviously you can damage your skin, and you can, in many cases, repair it, sometimes with a scar, but in general, it's pretty resilient. It's also defending you from infection, helps you regulate temperature, and in the lab, if that happens, I know right now it's kind of weird, but there's a lab towards the end of the semester where you would normally be able to do all kinds of temperature tests and stuff like that with your skin, and hopefully that will happen, but you basically have different types of temperature sensors in your skin for warm and cold. The synthesis of vitamin D, it's actually the first step in the synthesis of vitamin D. I mean, you can get vitamin D from your diet, but you can also make it. We'll talk about that really shortly. And obviously sensations, right? So your skin is able to, and I'm going to show you a few of these mechanisms, but your skin is able to tell the difference between light touch, tickle, pain, vibrations, pressure. These are all different types of sensors that you have in your skin to differentiate all of these different sensations. And of course, temperature, which we just talked about, okay? So when it comes to the synthesis of vitamin D, all right, so we always say that vitamin D is the sunshine vitamin, and most of us, living where we live, we're pretty low in vitamin D towards the end of the winter because we don't get very much sun. But actually, so your skin is actually helping you in the first step of making vitamin D. So the vitamin D that the final form is this one here, 1,25 dihydroxy vitamin D3, okay? But it's based on a cholesterol molecule. So it starts off, if you're making your own, like clearly you can get it from your diet. You can get D3 and D2 from your diet. But basically what you're going to do here is that you're going to take this dehydroxycholesterol. So if we break that down, right? So if you look on cholesterol, there's going to be a 7 hydroxy. So this is now hydroxynated, all right? So you've lost an OH group from that cholesterol. So that first step takes place in the skin. And this then gets converted to vitamin D3, this particular type of D3, cholecalciferol. And then this then goes through the bloodstream to the liver. Now, in the liver, this D3, as well as if you get from supplements, D2, is going to be converted into the 25-dehydroxy vitamin D3. And then the kidney is actually going to do the final step, right? And it will do so stimulated by parathyroid hormone. So in semester 2, if you're taking AMP2, you're going to learn a bit about the endocrine system. And parathyroid hormone is released when you have low blood calcium. And it does a number of things, but one of the things that it stimulates is this conversion, the final conversion to the final version of vitamin D. And the reason that's important is because you need vitamin D to absorb calcium from your diet. So if you're someone who takes calcium supplements, the best ones you can get are the ones that are mixed with vitamin D. In my mother, she's got osteoporosis, and she takes a large amount of calcium, but she always has to take the one with the D3 because that will help her absorb it. All right. Not that right now you need to know all these steps. As always, look at the study guide, but I just think it's kind of important because we talk about it and we talk about the sunshine vitamin. So this is to sort of break it down, and you can see it's actually a little bit more complicated than that. And you'll come back to this when you get to semester 2, all right, with the parathyroid hormone. And if you took 105, I know when I teach 105, I do the balance between parathyroid hormone and calcitriol. So just so you know, that's coming in semester 2. So if you were in 105, learning all of that is worth it. It will come back, all right? Now, when you're looking at the skin, there's a number of layers to it, all right? So there's only 2 layers. Very often you see these pictures with these 3 layers. This subcutaneous layer, or the hypodermis, that is actually not considered part of the skin. It helps to anchor the skin to the rest of the body. So either you can say the sub-Q or the hypodermis. That is, you can see, sort of fatty, so it's a mix of adipose and areolar connective tissue, which I'll talk about in a bit. But the skin proper is really just these 2 layers, the epidermis, so epi meaning outer or covering, right? And then the dermis, which is this layer here. So we're going to go through these different layers. And then, of course, you have your accessory structures, so hair, nails, and glands, and we're going to go over those as well. So why do we have multiple layers? Well, we'll see how we go through this, but each layer has a function, right? So the epidermis, this is really important for resisting damage and protection of the underlying tissues. And dermis is going to do your temperature regulation, you're preventing from dehydration. There's some healing that happens here. This is a lot of your nerve endings are there, and your hair and your nails all grow out from the dermis. So that's, so there's these different layers have these different functions. And then finally the hypodermis, insulating, storing fat, and anchoring the skin in place. But of course, remember, hypodermis is not considered part of the integument, all right? So it's just the epidermis and the dermis. All right, so now when we look at the epidermis, we're going to start there. So there's a number of layers to the epidermis, and you are actually responsible for learning those layers in order, all right? So we have 2 types of skin. We have thick skin and thin skin. So the very bottom, and I think it's easiest to learn this from the base or from the deep part. So the stratum basale or stratum basale, there is where you've got your stem cells, your melanocytes, and a few touch sensitive cells, and we'll go over those. The stem cells are going to be producing the main cell type, which are the keratinocytes. So the stem cells are going to differentiate into keratinocytes. Keratinocytes, they are the ones that pretty much make up your skin that you think of, and either mature now, like the stratum basale is just 1 layer. So right above that, you have this stratum spinosum. So that's where you have your living keratinocytes. They've got nuclei there, and they're making lots of keratin. So keratin is that intermediate filament. You also have dendritic cells or macrophage here that are really important for helping your immunity and keeping you healthy. You'll learn a little bit more about that in semester 2, about how that works and how your specific immune system works. But these macrophage cells are there. They've got a name called Langerhans cells. And then over the spinosum, and the reason they call it spinosum is because you're making keratin, and the keratin can sometimes look sort of granular, and then you've got these dendritic cells which are spiny. So it kind of looks sort of, like grainy. So that's why they call it the stratum spinosum. Then you have the stratum granulosum. So now you're really going to get these big granules of keratin here. And that is above the spinosum. And what's happening here is that the keratinocytes are starting to lose their nuclei. I just want to make sure I'm saying everything that's in my notes. Of course, I did it the other way around in my notes. Yeah, so stratum granulosum, we said keratinocytes producing lots of keratin. Now, here I said that I said in the spinosum, the keratinocytes are alive, and then in the granulosum, they're starting to die. So there's a few reasons for that. So first of all, as you're going up from the base, these cells are getting further and further from the blood supply, right? So they're getting less nutrition, less efficient gas exchange, but also these cells are so specialized to making keratin that they actually, I wouldn't say suffocate themselves, but they actually make so much keratin that also causes them to die. And here you're going to see the really large granules of keratin, so that's why we call it the granulosum, right? Down here you can start to see them in the spinosum, but it's the spininess, they call it, mostly because of these dendritic cells, I would imagine. Then we have this layer called the stratum lucidum. This layer looks kind of clear kind of thing, or shiny in some preps. Not in this one here. Wait, where is it? Oh, actually, it's not on this one. Lucidium. Oh yeah, there it is. Here it looks kind of sort of dark, but in some preps it will. So it's got like a shiny hue to it. It's like a different color. This lucidum. So this is thick skin. It's not found in thin skin. All right. So thick skin versus thin skin. Thick skin is found on the palms of your hands and on the soles of your feet. That's where you have that. But the rest of it, you won't have a stratum lucidum. And then the last one is the stratum corneum, which is very often the thickest layer. Here the cells, the keratinocytes are dead. So this is our dead layer. This is your protective layer. And you're losing these layers of skin all the time, because if you rub your hand on something, you're going to lose some skin cells. So what's happening here is that the stem cells, they're sitting on the basement membrane. Because remember, this is an epithelium, right? So they can't grow down. They can't grow downwards or deep. It can only go superficial. So as these cells divide, they're going to move upwards. And as they move upward, they get more and more mature, and then eventually they die out, all right? So it's this stratum basale that's going to, feed these other layers, all right? I'm just trying to think. Oh yeah, and then the stratum corneum, I'm looking at here, very often they call the cells here like bags of keratin. So they've got like a cell membrane and keratin and pretty much nothing else. And this is where you're really getting your protection, all right? Yes, okay. Now, so here you can see another view of that. Okay, you can see your layers, hopefully, and you'll see that the preps in the lab look more like this, but you can see there's these different preparations, and you can always see that stratum lucidium. It always sort of has a slightly different color. It's kind of weird. It's not going to be on this one because this is thin skin. All right, so here, palms and soles of feet. The other way is like the hairless skin. That's your thick skin. The other way of thinking of the one that is your thin skin is that's the hairy skin. All right, so yeah. Okay. Now, looking again, now here's a drawing where you can really see. So the idea here is that here in this stratum basale, this is where you've got your stem cells and all kinds of other cells that are sort of developing. The pictures that you see over here, they don't show you all the different cell types. So this is to remind you here in the stratum spinosum, you've got your dendritic cells, your macrophage, which are also called Langerhans cells. And then up here in the granulosum, you've got so much keratin that you can actually see granules of it. And then your lucidium, if you've got thick skin, sort of a different shade, right? So here again, you can sort of already tell that this is going to be lucidium here if you're paying attention. And then up here, corneum, all right? So you can see all these different preps. So you need to know them in order, right? So the easiest ones I think are easy, the basale, because it's the basement or the basal layer, and the corneum, even if you think of your eye, the cornea, most of you know that, in front of your lens, you've got the cornea, so that, that's outside in a way. And then in between, so lucidium, you'll know is going to be right underneath that corneum and only in thick skin. And then think about it logically, right? So, you're going to have the spinosum, you're going to have active cells because here active cells being producing the keratin. And you'll remember spinosum because your macrophage have to be in this layer here. And if they go too far, they need the blood supply, right? You don't want to kill your macrophage. So it makes sense that they should be right above your basal layer. And then the last one left is the granulosum. So remember that the keratinocytes, they start to die out, right? Why? Because they're producing lots of keratin. So that would make sense that the granulosum layer, because of those big granules of keratin, would be right underneath the cornea, all right? So unless you're in thick skin, which you would also have the lucidium in between, all right? So, so try to think about it logically, and then you'll be fine, all right? Okay. Now, I'm just make sure I'm not missing anything in my notes. Thick skin, thin, I'm on page 3. Increase in keratin, increased distance from blood supply equals decrease in viable cells and decreased number of nuclei. The most superficial layers are often just bags of keratin. All right, so now let's talk about the cells themselves, all right? So the cells of the epidermis. So what's happening here, remember that you're going to be replacing cells from the basement layer. So that's where your stem cells are. So you've got your stem cells in your basement layer. There's some keratinocytes in the stratum. The keratinocytes will still be dividing a little bit in the spinosum layer, and they're also producing keratin. In the stratum granulosum, they're going to start going through apoptosis and dying out, right? And then you're going to get vesicles in the stratum spinosum is going to start releasing lipid. That's the one thing I didn't mention. So that's going to also start to give you a bit of an oily feel on your skin, protect you from dehydration. And your epidermal water barrier is going to be between the granulosum and the spinosum, right? So that's going to help you prevent from dehydration. Now, the reason I'm showing over here is because the keratinocytes are connected by desmosomal structures. So if you remember back in our discussion of the junctions between cells, they're held together with cadherins, and they're going to link to these intermediate filaments. So in this case, since we're in skin, the intermediate filaments are going to be keratin. So this is a way of the keratinocytes of connecting together. So even once they die out, these connections will still be there, and giving you a good barrier for your skin there, all right? So if we look back, right, so this should also make sense to you that these you know, some lipid is going to be released here in the spinosum, because if we go back, remember the spinosum layer, whoops, I'm going in the wrong direction here. So remember the spinosum layer, I am going in the right direction. So you remember your spinosum layer, it's going to be right here, right? We've still got viable cells, they're producing keratin, and they're also going to release some of that lipid to help waterproof as well, okay? Okay, so this is your waterproof layer. All right. So here you've got the first cell type that we're talking about are the keratinocytes. So the keratinocytes, as you know, they're producing the keratin, and they are most of the cells there. So they're, you know, the keratin is going to help waterproof and help strengthen your skin. And then of course you've got a little bit of a lipid there that's going to help that as well. And these are going to be replaced by the stem cells in stratum basale, because these are the ones that are going to die out and form that protective layer in your stratum corneum, all right? Now, of course, many of you know about these guys, the melanocytes, right? So the melanocyte layer, and it's not a layer, but the melanocytes are living amongst the keratinocytes, especially down in the spinosum layer. And they sort of, you know, migrate around in here, and they're producing melanin, all right? Of course, that's what melanocytes do, all right? So we're going to talk a little bit more about those melanocytes, but there's, you know, the amount of melanin that these guys are going to produce is going to be dependent on environmental factors and also some genetics, right? And then as they're producing their melanin, the keratinocytes are going to absorb that, and that gives your skin its color. Now, the thing to remember is that there are 64 genes that code for skin color. So the amount of melanin that's produced is really going to, obviously affect the type of color that you have. Now, the thing that you might not realize is that even someone as pasty as I am is making melanin, all right? So the point is that there's more than one kind of melanin. I mean, most of the time we're thinking of that, the beautiful brown melanin that makes us look healthy, right? And let me go over here. All right, so here, so that's what's called eumelanin, all right? And, the amount of that will affect how dark your skin is. But someone like me, I won't make as much eumelanin. I make some, particularly when I'm exposed to sunlight. But I make a lot of this one here. This is pheomelanin. And pheomelanin has a different hue. It has sort of a reddish or a pinkish yellow hue. And depending on how much of this you make will also add that hue to your skin tone, all right? Now, interestingly enough, it's the eumelanin. This is the one that's going to be protected from the damaging UV light from the sun. So that's why we tan in the summer, right? So no matter how dark you are, you're still going to tan, right? I used to have a really good friend from Trinidad, and she used to get upset with me because she was tanning in the summer, and I was like trying to get browner, and she was like, I don't want to be more brown. It was really funny. So everyone's tanning, everyone's going to make more eumelanin in UV light, right? And conversely to that, I used to know some of my neighbors who would never go out in the sun, no matter how white they were, because they didn't want to have any sort of eumelanin in their pigmentation. And wow, I used to have a neighbor. She was oh wow, she didn't look healthy in any way, shape, or form. But so the point being there is that eumelanin we make because we're exposed to sunlight. But this other one is pheomelanin. It doesn't help you at all when you're exposed to sunlight. All right, so this one has that red-yellow color. It really absorbs UV in a really weak way, right? Now, why would we make it anyways? I mean, it's kind of weird, but if you look at my notes, there's one of them is an antioxidant and the other one is an oxidant. So eumelanin is an antioxidant, which makes sense because if you're exposed to UV light, you're going to be making free radicals, which are damaging to your skin. So it's also going to protect you, not only by absorbing that UV light, protecting you, but also getting rid of your free radicals, right? Whereas pheomelanin, it's going to be an oxidant, and that's going to be helpful for other purposes in the body, which we don't really discuss here, okay? But it's much more helpful for someone who doesn't get very much sunlight. So they have very, very different functions, all right? So that's my discussion of melanocytes. Now, the other issue, tanning, all right? So when you tan, so we were just talking about tanning, and we know that you get that beautiful dark hues because of the eumelanin. Now, some people say, oh, it's because my melanocytes are making more melanocytes. You're not making more melanocytes. The only thing that's happening is that your melanocytes are getting more active. I mean, there might be, to a certain extent, you might make more melanocytes, but in general, what's really happening is that the melanocytes you have are just more active, and you're producing more of that eumelanin, all right? Now, when the melanocytes do divide, obviously they must divide to a certain extent because they have to replenish themselves. But if you're having a problem and you're making too many melanocytes, that's when you get into cancer. So we're going to talk about that in a little bit. All right. Okay. So I think that. The other thing that adds color to your skin, we're going to talk a little bit more about that. Is hemoglobin, right? So blood, right? You got that reddish hue from the iron in the hemoglobin. That can also add to your skin color depending on how close your blood vessels are coming to the surface. And also keratin. This is kind of an interesting thing. I don't know if oranges produce keratin, but that's something else in the diet. But carrots, this is how the story goes. Obviously, I didn't see this because it was me, but and I was a baby. But when I'm born in March, I'm a March baby, and back when I was in my mom in my mom's tummy, she, there wasn't a lot of fruit for her to eat. And she had lost a lot of babies already. So she was eating oranges like crazy when she was pregnant. And apparently I was born with a color to me. Obviously it wasn't due to melanin, eumelanin, but I had like a healthy glow to my skin. And I'm guessing that that was due to some sort of, I'm not sure if it was carotene, but probably something in the oranges that my mom was eating really gave me this healthy hue. And apparently when I was leaving the hospital with my mom, everybody was saying, oh, look at your baby, looks so healthy, you know, but it was because of this color. So there's other things that can also affect your skin color. So that's, wanted to bring that up. And that you can get from your diet, right? And we'll talk about a few other ones in terms of diagnosis of, how well you are, your wellness factors, all right, can also be determined by looking at your skin. Now, let's go back here to the different cell types. Oh, I'm going the wrong way. I'm sorry. I'm going to completely mess you guys up. Let's go back. All right, so melanocytes. So the next things are the dendritic cells or the Langerhans cells. And these guys are, like I said, they're going to migrate around in the spinosum. And they're looking for debris and, bacteria and stuff like that to clean up from your skins, right? Because, if you cut your skin or you get some dust in your skin or something, these guys are there to clean that up. And they also, like I said, you'll learn about this in semester 2 a little bit when you learn about specific immunity. But they also have a way of processing what they ingest by phagocytosis and presenting that to your immune system. So that way you can alert your immune system if you have an infection, if you're a bacterial infection, your immune system will start to kick in and treat that for you, all right? So these cells are called, technically they're called Langerhans cells, but they are dendritic cells, which is a type of macrophage specifically geared to your immune system. Now, this last type of cell is the Merkel cell. And the Merkel cell is a tactile cell. So it's a sensory cell. It's not a neuron, all right? But it has the ability to sense touch. And it says here, it sends that information to the brain. Well, since it's not a neuron, it really can't do it on their own. So these cells transmit by neurotransmitter once they're stimulated due to touch. They release a neurotransmitter to a sensory neuron that is associated with them. And this neuron is actually not in the epidermis, it's in the dermis, all right? So the Merkel cell is in the epidermis and the neuron is in the dermis, all right? So these two work together, but the Merkel cells, like I said, are found in the epidermis, all right? So if we're looking at this, so where are you finding the Merkel cells? So the Merkel cells are going to be in the stratum basale with the stem cells, okay? You're also going to find the melanocytes here, and they're going to sort of reach out and be releasing their melanins. So whichever ones they produce and how much of eumelanin or pheomelanin you produce is dependent on your genetics, and then your exposure to sunlight. And then, of course, then your keratinocytes are going to be a little bit higher up, because down here you've got the stem cells, and the keratinocytes are going to be developing as we go up in layers. All right. Okay, so let's go through this, and I already went through this. So we've got where we're making our keratin and how we're getting our water barrier and our connections between our keratinocytes to make this cohesive layer of skin. We went over our melanins and the different types. I wanted to show you this too. Not that I'm going to test you on this, but I want you to see the structure of these different melanins. And you can even see with pheomelanin, there's more than one type, and eumelanin, there's more than one type. And you can already see that eumelanin and pheomelanin have different structures, right? All right, so here you can see that here you've got a 6-membered ring and a 5-membered ring, right? And there's some little add-ons here, but this is eumelanin. And then pheomelanin also has these ring structures, but then you've got these different other, things hanging out here. The thing that you're going to see that's in common is that they're rings and double bonds. And it just so happens that pigments in general have rings and double bonds. And then these functional groups are going to make them different in terms of what they're named, all right? But this is just for your information so that you can have a bit of an appreciation as to how this works. Now here, what I wanted to show you, I love this picture because here you can actually see a Merkel cell, all right? So remember, this is your sensory cell, right? So here they draw it like this, and then they have the associated neuron here in the dermis. But this is what it actually looks like. So you can actually see them differently. These are from, in the stratum basale, right? So here you've got your stem cells. And here's this cell that has a little bit of a halo around it. So it just so happened that this is a really nice cut through the epidermis. And you can see they caught a Merkel cell here, all right? Okay, so now we're going to talk about the dermis. So I'm going to look quickly at my notes to make sure I'm not missing anything important. Oh, when you're talking about these Merkel cells, notice how they look sort of roundish, right? So sometimes if you depending on what book you're looking at, they might be called the Merkel disc, all right? And that's why, because they have this kind of like sort of oval sort of disc shape. So that's a name that will sometimes be associated with that. Okay, so I think that's everything that's in my notes. Okay, now let's look at the dermis. So that's the next layer. So remember, your epidermis is your epithelium. So if you remember your tissues from our tissue chapter, which chapter was that? Chapter 4. Any epithelium has got to be associated with a connective tissue. So your dermis is considered your connective tissue layer. And with that, they divide it into 2 sections. So there's the papillary region, okay, which is up here, and then the reticular region. So the papillary region, you can sort of see, is where we've got these ridges, and these are often called the dermal papillae, all right? So this is the one that's more superficial. Now, if you look at the types of protein fibers that are there, you've got loose areolar connective tissue. So you've got collagen and elastic fibers. I wrote that in my notes. Here you've got a lot of free nerve endings as well to help you detect all of these different sensations. So we'll go over some of those as well. So we've got free nerve endings to detect, tickle and pinpricks and temperature. And then you've got these other structures called Meissner's corpuscles, which are better at detecting deep touch, and Pacinian corpuscles, which are better at detecting pressure, right? So the difference between that is a pretty fine line, I guess, but you've got different sensors for that. And the papillae are these bumps. Now, why do you've got these papillae, right? So remember your epidermis, it doesn't have a blood supply. So the idea here, the idea that, is usually written in books, is that the papillae allow these nerve endings and the blood to get a little bit into the epidermal region, right? Not that actually penetrating, but the bumps are kind of penetrating it, right? And that's sort of the idea here. And you'll notice that a lot of the free nerve endings are sort of, drawn like in these little ridges, right? And the interesting thing is that these ridges give you your, if you look at your skin, you can see that your skin has sort of texture to it, and especially on your fingertips, right? So those ridges are extending into your fingertips. I'm actually looking at mine right now while I'm talking about it. So these epidermal ridges, or these papillae, are reflecting the contours in these, in the papillae, right? So they're going to form the basis of your fingerprints and, any markings you have on your feet as well. And why do we have those? Well, it's not so that police can identify you. I mean, those are there so that you can get a firm grip on stuff. That's why you've got really prominent ones on your fingertips, right? And there's some people that actually don't even have fingertips. They have fingertips. They don't have those ridges, which is really kind of weird. So when they take fingerprints, nothing will show up there. I was actually watching a movie once where they actually did that to somebody, got rid of their fingerprints so that they wouldn't be able to be identified. But anyways, that's movies. Anyhow, then you have your reticular region. Now here you're going to have dense, irregular connective tissues. So now if you remember that, if you remember your pictures of dense, irregular connective tissues, that should bring to mind these weird, chunky pieces of collagen, right? And there's going to be some elastic fibers there. And you're also going to start getting some adipose cells, right? So that's going to be right above your subcutaneous region, right? Or your hypodermis, where you have lots of adipose cells, all right? So there's going to be some in the reticular region. There aren't any in the papillary region, at least they're not normally, okay? Now, also within the reticular region is where your accessory structures are really growing from. So remember what those are, your hairs, nails, and your glands, all right? So in my notes, first I go through hair. So let's get to that, because I know there's a few more slides here. So this slide, the purpose here isn't so that you start memorizing every type of neural sensational kind of thing in your dermis. It's to start to give you an appreciation of those, right? And when we do this in the lab, there's even more. If you lab with me, I have a slide with a whole bunch of different types of them. But the ones that you probably want to learn the most, that understand that you have some that are free nerve endings, and they are going to be detecting all kinds of things like temperature and different types of sensations, all right? The Merkel discs, so remember the discs themselves are right into the discs themselves are in the basal layer, and then the neuron is associated with that in the dermis. And then the Pacinian corpuscles are pressure, and Meissner's corpuscles, they're going to detect touch. And then they sort of differentiate them here because we can see touch a little bit closer to the epidermis, whereas the Pacinian corpuscles are a little deeper and they're going to be detecting the pressure. And your brain is able to differentiate between those. There's also Ruffini endings. Those are also detecting pressure. I don't know how that's different pressure from this. I don't know enough about it. I just know that there's also this one. And then you're also going to have a neuron associated with your hair. And those are in there's a plexus of neurons here. And that's how you know when someone pulls out your hair, because your nerve feels that. And if you just sort of brush your hand over the hairs in your skin, you're not touching your skin, you're touching your hairs, but you can feel that, right? So I'm doing that right now, actually. So you can give that a try, but that information is being sent by your root hair plexus to your brain, that someone is sort of stroking the outer layer of your skin without touching your skin, just touching your hairs. And when do this is going to give you a weird sensation, but when do I notice that the most? If I walk into a spider web out on my porch right now, right? So it's the end of the summer, so that's the time of the insects, right? It's the early summer, it's time of birds, and in the summer, insects, and you walk into a spider web, and I can feel that with my hairs. It's definitely that feeling. If I'm doing that right now to my skin, it's like, oh, that's what I feel when I walk into a spider web. Just a thought, all right? Now, if you look at your hair, there's some basic structures that one should know, right? So when you do this in lab, you're going to learn more in lab about this. And there's always this fine line between learning the parts of the hair and learning the parts of the nail. Typically, what one does in AMP1 is focus on the hair. So if you're going to spend time on something, work on the hair. In my notes, you'll see that I've got different types of hair. So languinal hair, vellus, and terminal hairs. So languinal, that's fine non-pigmented hairs from the fetus. So that's what the babies have all over them, that soft hair, which they lose. Then the vellus hair are the short, fine hairs that you have all over your body before puberty. And then the terminal ones are those coarse, pigmented hairs that you get on your body, you know, once you're mature, all right? So they're just slightly different types of hair. And here you can see vellus hair and terminal hair. Now, in terms of the parts, so remember you've got a nerve or root hair plexus. If you're in my lab, there's a part of the lab homework where you're actually going to draw out all the different parts of the hair. So I recommend that. Then there's a muscle attached to the hair itself, and that is called the erector pili muscle or the piloerector muscle, depending on who you read. It's the same thing. It's a smooth muscle, and it's the one that pulls your hair and makes it stand on end when you're cold, right? So the idea here is to trap a warm layer of air above your skin to help keep you warm when it's cold. Now, your genetics is going to determine the thickness and the distribution as well as the color of your hair. So again, you're going to have a mixture of pheomelanin and eumelanin in there. And then as you get older, your cells just stop producing melanin, and then your hair gradually goes from gray to white, which I'm getting more of right now. And my mother's hair is completely white. Yeah, another story I could tell you, but that makes these videos even longer. Then of course you've got the different parts. So here you've got nerve fibers. So then you've got the follicle, right? So we're going to go over this a little bit. You've got different layers, and it's best to just show this with a picture, and then I can combine that with my notes. And then the overall structure. So, you know, the bulb and the root and the shaft, okay? So if you look on my notes on page 6, all right? So shaft is the hair above the surface of the skin. So up here is the shaft, okay? The follicle is anything below skin level. So when we're talking about skin level, below the epidermis, okay? So below the surface here. So that is now the follicle. And then the root itself, that's the part that's really penetrating here in the dermis, deep into the dermis, and that's where you've got your stem cells and all of the nerves and the blood supply, all right? Okay, now in terms of the layers, so if you're looking on page 6, I have these layers organized from superficial to deep, all right? And this picture, I think, is the best one to show this. But before I get into that, right, so you can see the different, and I know this is a lousy picture, but you can still get the meaning from it. So the darker your hair, the more eumelanin that your melanocytes are producing into your hair. The blonder you are, the more pheomelanin and eumelanin. And then someone, like when I was younger, I had sort of in between hair, so I had a mix of that. So more pheomelanin and a little bit of eumelanin. This is a little bit more red than I had. I was a little bit more like a light brown. Actually, I was blonde until I was like 3. And then I guess my, you know, eumelanin producing melanocytes got, you know, kicked in, and then my hair went dark. But anyhow, the other thing before we get into these layers that I want to draw your attention to, this was something I used to love to do in the lab. I don't do it now, but I used to love to have a slide where I had a cross section of hair. This is like looking down from above, and it's cutting it right through. It looks very different than the side view, right? And the reason I did this is because some students would get it confused with a gland. So the reason you know this is not a gland, first of all, it's got way too many layers, all right? So a gland does not have this many layers. But the other dead giveaway is that you'll have the pigment in the middle here. A gland will not have that, all right? Most of the time though, and I think the slides that we have in lab show the hairs from the side, so you can see that. And here you can also see your sebaceous gland next to that. I'm going to get into that in a sec. I'm going to talk about glands. But the sebaceous gland, it's really obvious once you know what to look for, and I will explain that to you. Now, here you're looking at the layers. Now, these layers, this is crazy, right? If you're going to learn layers, learn the layers of your skin first, all right? Because I've never asked this, the different layers of the hair. But it's in your learning outcomes, so I'm going to go over it with you, all right? So if you go superficial to deep, all right? So you have first the dermal root sheath. And of course, outside of that, you also have a connective root sheath. There's connective tissue out here, all right? So you can see there's lots of connective tissue out here. So there's a connective tissue root sheath that's holding this all together. But below that, you have the dermal root sheath. And then you have this layer called the epithelial root sheath, and that's made up of 2 layers. It's easiest to see this in my notes. One called the external root sheath, which is going to be the more superficial here. And then the internal root sheath, which is deeper. And then you have your layer of cuticle, right? So then this is like now deeper, so you've got your cuticle on this layer here. And then deep to that, you've got your cortex. And medulla is always in the middle, all right? And medulla, always, always, always in the middle. And cortex is always outside of medulla. So hopefully that will help you, right? You know, memorizing these layers, I don't know how that's going to make you a better physiologist. But there you have it. And it's always part of the learning outcomes. So I wouldn't kill myself over this. I just think that, you know, you should learn it. You can draw it out for lab if you have my lab. Don't forget the other things, your capillaries, your blood vessels here. And notice your melanocytes are down here. So they're the ones that are going to be producing that melanin. So here's like, just like you had with your skin, it's very, very similar. So you got this basal layer with your stem cells here. These are the mature stem cells. I'm going to get into that in a sec. And then you've got your melanocytes, and they're going to be producing the melanin, and that's going to be going up through the core here into the medulla. And that's why I was saying that you've got your pigment here in the medulla, all right? And that will get dispersed out depending on how much pigment you're making. But eventually, above this, these cells are all dead. There's no living, viable cells up here. Okay, so your hair is basically dead keratinocytes and protein. All right, mixed with some pigment. And I know I wrote that down there. Oh yeah, actually on page 5, dead keratinocyzed epithelial cells here. Okay. Now, the next thing, I'm supposed to sort of go over the growth cycle of your hair. And you probably notice that certain times you have hair that falls out more frequently than others. Very often that's hormonal. But there's a whole science behind this. And if you're interested in this, it's really cool. There's I did a postdoc at the Rockefeller University. And there's one of the, not with this woman, Elaine Fuchs, but she's also there. But she studies stem cells. And the way she studies them is she looks at hair. And she studies stem cells in that way. So basically you have your stem cells, and this is kind of the weird thing. You might think that your stem cells are here in the bulb, right, in the very bottom of the bulb, and they are, but not your really important stem cells. So you've got a set of like really primordial early stem cells that self-renew themselves. They're actually found above that in this area called the bulge. And this is where your stem cells renew themselves. And then when they become mature, right, they're actually going to go down to this bulb and they're attracted by the blood supply. I guess that's the hypothesis. And then they're going to start making the keratinocytes and everything, as you know. Now, this is important that they're not found down here. And the reason that's important, and what you're looking at here is, you know, work from Elaine Fuchs' lab. You can see that this green here, these are the stem cells. They're stained with a certain antibody to them, and that has a fluorescent marker attached to it. And they reside up here, okay? And then they go down here and do their work, all right? Now, why is that important? Well, basically what happens is that your hair follicle goes through a cycle, and it's responding to different, you know, factors in your body. But every hair follicle will do this. It goes through a growth phase where the cells are going to divide, and you can see that as it does that, you're going to extend your bulb, and it goes down, and then you go into this regression phase where they're going to stop dividing, and they're just going to hold on to the hair. And then you're going to get to a point where they're going to rest. And then what will happen is that your hair will fall out and the bulb will shrink back, and then you're going to get back up to this bulge here, right? So your hair, each of your hair follicles goes through this cycle, just so you know. So it's really important that the stem cells proper are actually not down here in the bulb where your hair is growing from. They're a little bit higher, and you know, they'll you know, cycle through as needed as your hair goes through its growth cycle. All right. Yeah, something you might not have known before. I found it kind of interesting. But anyways, if you want to learn more about it, you could probably follow this link here, this Nature article, and hopefully you can get access to it, and then you can sort of learn a little bit more about it. Anyhow, okay, so that's what's happening with your hair. So nails. So you'll see that there's a very similar sort of pattern with the nails in terms of having where you've got your stem cells and that sort of thing. I wouldn't belabor this, all right? With this slide, this actually I got from Dr. Tice. So you can see here it actually divides the nail into these different parts, right? So you've got the nail bed, where the skin on which the nail plate rests. So what's the nail plate? Well, the nail plate is the thing that you are familiar with, right? You've got your free edge, your nail body, and then where you've got your cuticle here, right? So that's your nail plate. And here it sort of tells you what that is, right? So here you've got this clear keratinized portion of the nail. Now, the other part here, the hypochaeum, this is where you've got your nail bed, right? And then in that you've got your nail fold and your nail groove, right? So you're, where the fold around the margins of your nail plate, and the groove is where you get your annoying hangnails, right? And then here, your nail matrix, that's where your nail, where you're growing from, right? And the reason I don't love this picture, that's why I included my other one, but here you can see your nail matrix. It's going to be here. So that's where your stem cells are going to be, that are going to be producing your keratinized cells, and then they're going to grow out, right? And then they're going to rest on the bed. So these cells in the matrix, they're the ones that are going to be dividing and producing the cells that eventually will make up this. But of course here, the cells are no longer living, similar to your hair or the stratum corneum of your skin, right? So those are dead keratinocytes. I like this picture a little bit better so you can see things a little bit in a different perspective. It's hard to find one picture that has everything that I want in it. And then here you can also see your phalange, so your phalange from your bones, right? So you're going to learn that later in the semester. But like I said, I would not kill myself over this. Focus on the study guide. Just know what's on the study guide. Usually, like I said, in AMP courses, they focus on this one, all right? Which will be really important if you end up taking, if you're doing AMP2 with us, that's at Morrisville, that's fine. If you're going to transfer to a different school and you do AMP2 somewhere else, I would really lean towards learning my hair parts, all right? Okay, now glands. So that, again, part of the dermis. So I really want to draw your attention to the chart I have on page 7 of my notes, all right? So overall, you have 2 types of glands, and I'm going to start off with the easy one, which is the oil glands, all right? So the oil glands are also known as sebaceous glands, right? So they're producing sebum, and all of those drain into your hair follicle, all right? So I'm going to show you some pictures of that, but I can go back to the picture I already showed, right? I said this was really obvious. Because this, to me, when you see this looks like drops of oil to me, all right? Maybe it's just me, but, the sebaceous glands always have this sort of like bubbly look to them. And to me, it's oil, right? So I know that that's my sebaceous gland. And always when you've got a sebaceous gland, you're close to a hair follicle because this oil drains into the hair follicle and then it comes up to the surface of the skin. All right. So that's the easy one, right? So here you can see in the diagram, sebaceous gland. Now, the hard part are the sweat glands, all right? So the sweat glands, they're also called sudiferous glands. And you have 2 types, all right? You have the eccrine sudiferous gland or the eccrine sweat gland. And most of your, as an adult, most of your sweat glands are those. And they do not drain into the hair follicle. They drain to the cell surface, to the surface of the skin directly, all right? So if you're looking at the models in lab and you'll see this sort of duct going out to the surface, then you know right away that's an eccrine sweat gland, all right? Now, and then if you see a gland draining into the hair follicle, that's your sebaceous gland, all right? Now, in the lab, we do not have, and I have to look at the models again, but there are no apocrine sweat glands on those models as far as I know, all right? Now, apocrine sweat glands, well, how are they different? Well, they're draining into the hair follicle, but only in certain places, all right? So those would be your stinky places, right? So your armpits, your perineum, all right? So your perineum, what is that? Well, that's that area around your genitals, right? So between your male parts or your female parts and your butt, right? So that's your perineum, pretty much, right? And then the other place that you'll find that particular type of sweat gland are around your nipple, right? So they call it the areolar region of the breast. So that dark area around your nipple. That's the other place you're going to find those. All right. Now, the other weird thing is that those apocrine sweat glands are the same ones that become mammary glands in the breast, so for lactation, and your ceruminous glands. I always have trouble saying that, in your ears, producing earwax. All right. So those apocrine glands are sort of the stinky, gooey glands that we don't like to think of, unless you're thinking of mammary glands, because you like to think of, you know, making milk. That's good for baby, right? But they are modified sweat glands, and specifically modified apocrine glands. Now, the really weird thing, okay, is that infants, all right, so the infants, they don't have eccrine sweat glands. So they don't have those glands that drain onto the surface of the skin. Oddly enough, they have apocrine sweat glands. So baby sweat and adult sweat is different. The nice thing about baby sweat is it doesn't smell like grown-up sweat, right? Babies smell nice, especially if you put baby powder on them, right? But so that's kind of a weird thing. But in the infant, the glands are apocrine, and it's kind of weird, right? But FYI, and then as the the baby grows up, they'll stop producing that kind of sweat gland, and they'll produce the eccrine sweat glands. Just so you know, all right. Okay, so I think that's everything. So have a look on page 7. I think that really simplifies things. Looking at that chart that I tried to put together here for you. All right. So here is the slide that goes with that. So the eccrine versus the apocrine sweat glands, right? So apocrine drains into the hair follicle, but only in the stinky places, and around your nipples, in your, and the weird ones that produce wax in your ears and milk in your breasts, all right? Oh, the other little tidbit here, the eccrine sweat glands that drain out onto the surface, sometimes they're called merocrine glands, just to make it more confusing. But if you go back to the way glands secrete, and you'll see that there's the merocrine secretion, right? So they're named after the way they secrete sweat, right? So that's basically the idea here. But I think we're going with eccrine in this course. So if you see merocrine in your book, whatever book you're using, this is the same thing. Eccrine sweat gland and merocrine sweat glands are the same, all right? Okay, great. So here's some pictures. So here again, I know right away that this is a sebaceous gland because this looks like oil to me, right? So I know this associated structure has got to be a hair follicle, and now you can sort of recognize it. Here, you know that this is going to be your eccrine or merocrine sweat glands because it's draining to the surface. And I don't know how you're going to know that this is apocrine, but this apparently is apocrine. I guess the best way is I would try to landmark it around if you know where the tissue is located from, or if you see a hair follicle, neither of which you see here. But anyways, this would be an apocrine gland. But these ones are easy, all right? Hopefully that will help you. This is more to help you in lab, all right? So I would never do this to you in lab, and I don't even know if we have apocrine glands in our histological sections. By the way, in the library at Morrisville, we have all of the slides there. So I just want you to know that. That might be useful for your studying purposes. Now, the hypodermis, that lies beneath the skin, right? So remember, that's your subcutaneous layer. Sometimes you'll hear it referred to as the subcutaneous fat because it has lots of adipocytes, all right? So its function then is to attach the skin to the underlying organs. And you've got nerves. You've got some more pressure receptors there. That's what these are here. So these are, another type of your Pacinian corpuscles. They are also going to help you really detect deep, deep pressure, right? If these ones are stimulated, because it's all the way below your dermis, all right? What else can I tell you? I think that's about it. All right. So now we're going to go into some other little variations. So I had said to you that there's like your skin color can tell you a lot about yourself. And when I'm talking about that, I'm not talking about your normal skin color, but odd skin colors, right? So here's a list of them. So cyanosis is if you've got sort of a blueness to your skin, which would indicate oxygen deficiency. Erythema is redness due to increased blood flow to the skin. So if you blush, that would be, if you're blushing because you're embarrassed, that would be like an erythema. Pallor is paleness, and that's a decreased blood flow. Albinism, I think I wrote some notes on this. Yeah, so complete or partial loss of pigment in the skin, hair, and eyes. It's largely genetic, and it's a result of a defect in the enzyme needed to produce melanin. So this is any kind of melanin. So this is really considered devoid of color, right? So this is the difference when you're not producing any melanin, right? So you're not producing pheomelanin, you're not producing eumelanin. So if you ever see someone who's truly albino, it will really throw you. I saw someone like that in the library once, and it was amazing. I was just so it was just so interesting. But anyway, so albinism. Vitiligo. Oh, when I say the library, I mean my community library. I don't mean Morrisville. Then we have vitiligo, which is pictured here, right? So this, we've got patches of depigmentation. I actually don't know why that happens. For some reason, I guess the melanocytes in those areas, for some reason, are having trouble producing melanin. I'm not sure why. Jaundice, yellowing of the skin, and that's because of bilirubin in the blood. So bilirubin is a pigment that has really yellowy color, and it's a byproduct of the breakdown of hemoglobin. And this is often associated with a problem of liver function, and even the eyes will turn yellow if you've really got severe jaundice. Hematoma, that's just the AMP term for a bruise, right? So clotted blood under the skin, all right? Then we have these other things. I absolutely do not expect you to learn all of these, types of skin lesion cheat sheet. I'm not expecting you to learn this. This is here for your reference, so you can see all of these different terms. And who knows, if you're getting into healthcare, if you end up working for a dermatologist, then you're gonna have to know all these things, right? Or if you end up becoming a dermatologist, I'm making an assumption there. Because I know a lot of you are in the nursing program. But I used to have a good friend when I was an undergrad. She was an RN, and then she went to med school. So who knows what's in your future. But here are all of the different terms for all of these skin lesions. The other things, warts, nail fungus, cold sores, I didn't even get into here, but there's other ones. Ones that I do want to talk about is callus. So a callus, if you have an area where you have lots of friction, your stem cells will cause a production of keratinocytes, and that will be there for protection. And that can become a corn if it's really localized. Dandruff, the loss of keratinocytes from the scalp. And then there's a whole thing, the difference between a freckle and a mole, right? So freckle, usually lighter in color than the mole. A freckle, those will fade. Usually it's because of sunlight exposure, whereas a mole is definitely permanent. You're always going to see it, right? The freckle is flat and a mole isn't. So those are the big differences. Then there's the hemangioma. So at the top of page 9, I have a little note on that. It sounds like a cancer because it has that oma in there. It's not. But basically, we say it's a non-cancerous growth of blood vessels, and you have two different types. So you have a capillary hemangioma. That would be just proliferation of the small blood vessels. And then a cavernous, which is a proliferation of large blood vessels in the area, all right? So you guys will call it the capillary, right? In Canada, we call it the capillary. So yeah, no, I didn't say it wrong. I just said it the way we say it where I grew up. Now, the other thing, nevi, I don't have a picture of that, but a nevi very often is considered a mole, but it's really a birthmark. So if you have some sort of a mark from birth, they'll call it a nevi, and usually we think of it as being a mole. But it can be different types of things. Like I have myself, my birthmark, I guess it's sort of like a capillary hemangioma. It's underneath my hair. I've got like a patch of pinkishness, and it's always there. So would that be a nevi? I don't know. But the nevi usually is a birthmark or a mole that you have at birth. And then we already talked about the friction ridges, what we have those for. And there you can see the fingerprint, all right? Now, really important, skin cancer, close to my heart. Why? Well, because I used to study that as a graduate student. My doctorate I was studying melanoma. But the point here is that usually when people think of skin cancer, they think of melanoma. But there's different kinds of skin cancers. Very often they are triggered by exposure to UV light from the sun, but there's different types. You can see here basal cell carcinoma. So that would be those stem cells. Then you have a squamous cell carcinoma. That would be the keratinocytes. And then malignant melanoma, that's the one we usually think of, right? So that would be cancer specifically of the melanocytes, all right? This one, though, it's curable, but you have to be vigilant, which is why we talk about the ABCDE rule. This doesn't have the E there. I snagged this slide as well from Dr. Tice. But it's actually the ABCDEs of melanoma, which are here. Okay, so this is sort of giving you an idea of what this is. So ordinary mole versus maybe a melanoma, right? So asymmetrical, lacking of a border, changing color, a diameter bigger than 1/4 of an inch, a changing color and sizes, right? So, and if you've ever had your moles checked with dermatologists, I have, they take pictures of them. And then you go in again and they'll compare, different moles or different patches with your previous visit. So that's why they do that. I actually had a mole removed because I told you I studied melanoma. And there's this thing, if you're in graduate school or you're getting your PhD, they say very often you're going to die of the thing you're studying. So I'm like a little crazy. But I had a mole. And I went to the dermatologist. He says, I don't think it's a problem. And I went through my ABCDEs. I said, yes, but it's asymmetrical, it has a change in color, and it has evolved. And then he looked at me and he says, okay, I agree, let's get rid of it. And it's really easy to get rid of it. And then they send it for a biopsy, they treat it like a biopsy, and then they test it. So mine was okay. But if you know your ABCDEs and something is bothering you, and you know your skin better than anybody else, if you feel that something has changed, just go and get that thing removed, all right? It doesn't really hurt. They just put some dry ice on there and then they kill it and then they cut it out. So it's okay, right? I did it right in front of my daughter. She was young at that point. She was watching me, so I couldn't cry. No, you wouldn't cry. It doesn't hurt. It's a little uncomfortable when they take it out, but it's much better to have that peace of mind to know that that thing is not going to become a melanoma, all right? Now this is just to remind you about the stages here, because there's this whole idea of metastatic and all of that whatnot, right? So where does that come from? I've shown you this slide before, right? When you were learning your hyperplasia versus your dysplasia and all of that. So remember the difference here, and this is why I'm showing you. So dysplasia is when the hyperplasia starts to look weird. And then you're going to go on to your benign tumor. So here you got something that's growing, but it's still held in by the tissue margins. But a malignant melanoma, that's when it breaks through, right? Still not metastatic yet. But the malignant one is one that's going to break through. And where's it going to go? Well, it's going to love to go to a blood supply, right? And then if it gets into the blood supply, there is the possibility that it's going to be able to leave and grow somewhere else. And that's a metastatic melanoma, right? Or a metastatic tumor, right? So what's happening here, when you talk about malignant melanoma, it's really, really good at becoming metastatic. For some reason, melanocytes, when they become melanoma, are really good at growing in all kinds of places. And one of the jobs I used to have before teaching is that I worked in the Department of Neurosurgery at NYU, and we were talking about metastases to the brain, and very often the metastases were melanoma. Another one that metastasizes really well to the brain is breast cancer. Just so you know, but that's sort of where we're going with that. These benign tumors very often are called in situ because they're meaning in tissue, so they haven't broken out of the tissue, all right? Now burns. First, second, and third degree burns. And the easy way to remember that is how deep it goes, right? So if you're going to start superficial and go deep, and each degree of burn is one layer deeper, right? So first degree is only epidermis, second degree epidermis and some dermis. Third degree, you've damaged your epidermis and your dermis, and it can get into a full thickness burn, right down to the hypodermis. All right, so you can sort of see the differences. And what's happening here is that once you've gotten into the second and definitely into the third, is that you've damaged your stem cells, right? So this is going to cause severe scarring depending on how bad it is. And if you've really damaged it, the only way to give you some protection of skin again is to do skin grafts. So the full thickness burn is the third degree, right? So this is why you hear burn victims, if it, they say, second degree burns on 70% of their body or whatever, and you can sort of imagine what's happening there, okay? I used to do this, if you keep in mind, going back full circle about the functions of skin, what will happen to a burn patient in terms of the destructive damage of the skin, right? So temperature regulation, dehydration, fighting infection, all of these things get more and more complicated the more severe the burn and the more of the burn that's covering the body, all right? So here's another look at that. I like this one better because it sort of shows you it a little bit, more dramatically how to do that. Some of these slides where I'm doubling up, some of them I've snagged from Dr. Tice, and then some of them are my own, right? So now here, this video, this is a crash course video going over the integument. It's going to review everything. I recommend it. It's Hank doing it. So he's always entertaining. So hopefully that'll be helpful to you. The last slide is talking about wound healing. So wound healing, it occurs in 4 stages. I outlined them in my notes. So the first step is hemostasis, which is basically forming a clot. So what does that involve? And you'll learn about this a little bit later in semester 2. If you took 105, it's going to have the 3 steps. So vasoconstriction, platelet aggregation, and formation of the clot. So you're basically, healing up the wound. Then the second phase is inflammation, and that's the neutrophils and the macrophage going in there to clean up. Then you've got your proliferative phase. So your fibroblasts are going to go in and repair and lay down once again your fibers, so collagen, etc., that you're going to need. Get your matrix back. Your epithelial cells are going to start to proliferate, provided you haven't damaged your stem cells. And then you're going to have remodeling. How well that works will depend on how severe the damage was. And sometimes you'll have some scar tissue left over because you won't be able to remodel completely. Now, a lot of this work to learn how this is done was done in cell cultures. As you can sort of see here, the side view about how this was learned, right? So the stem cells are going to come in and reestablish the basal layer, and then they're eventually going to fill in the upper layers as your epidermis repairs itself. But what you can see here on this link, if you're interested, you can see one of the assays that are used for studying wound healing. And you can sort of see they score the cell culture, and then you can see the cells try to come back in and fill in the space. So this is how a lot of the initial understanding of wound healing was derived, and you can see a lot of, and this is the results of that. There are other people in the lab, they actually have really sophisticated models of skin that they grow in culture. And you can see why this is important, because we just talked about burn, right? So how can we treat someone? This is really obviously a very bad 3rd-degree burn, but it's still quite localized. If you have a burn victim who really needs grafts, and you need to understand how to repair these burns, for example, understanding how wound healing takes place is really a huge thing, because that way you can help them. But yes, there's a whole science behind that. But yeah, this is sort of a bit of a curiosity. One of my PhD advisors, he used to do this kind of work, so that's kind of why I threw that in here. But other than that, I think that's all the topics for the integument. Of course, there's more here than you're responsible for. But since you're in A&P, I'm sharing all of that with you. And again, you focus on the study guide so you know what you need to know for your test, all right? All right, so hopefully that was helpful.